Topic is about disentangling local adaptation drivers in the Andean tree species Nothofagus pumilio. So in other words, I'm an American who went to Germany and then went to Argentina to try to figure out how trees can live their entire life in one place. I'd like to say that I've learned something from this, but here I am in Switzerland. So the overarching point of my project is studying local adaptation in situ. So this means in a wild forest with a non-model species. And as we've seen in a couple presentations before this is kind of a hard thing to do because local adaptation is a very complex process. There's a lot of different factors that go into it, and they're kind of hard to pull apart. But luckily, there's a bunch of different ways that we can actually study those factors directly, either individually or in concert with each other. So in this graph, you kind of have the three main things that you can quantify for local adaptation. So you have genotype, environment, and phenotype. And for my project, I will eventually look at all three of these things, in addition to gene flow. However, for this presentation, I'll just focus on genotype, environment, and the combination thereof, which is environmental association analysis. So one way we can make it a little easier to study all of these factors and kind of pull them apart is to pick a good study system. And for this project, we went to the southern tip of South America, specifically the Andes mountain range. And the mountains in this part of the world are pretty cool because they have this really straight north to south orientation, so it creates a couple of really clear environmental gradients, one of which you can see from space. So this rain shadow effect you see here, this is mainly driven by the wind, which comes from the west, so it picks up all the moisture in the Pacific, hits the mountains, drops it, and then as you move west, it gets much drier. The other gradient we have is a latitudinally driven one, namely we have day length and temperature. But of course, you also need to study species that actually inhabits all of these gradients, or the point is kind of moot. So there's 7 different Nothofagus species in Argentina. Nothofagus pumilio is the most widespread and common. It grows all the way from 35 degrees south in a continuous line about 2,000 kilometers to Tierra del Fuego at 55 degrees south. It inhabits a lot of very heterogeneous environments as well. So in this picture, every single tree is a Nothofagus pumilio, from the very marshy area in the valley where I'm standing all the way up into the foothills and to the tree line. And it's a cold-adapted species, so it often forms natural monospecific stands. In addition to all of these kind of heterogeneous normal environmental conditions, there's a lot of short-term stressor events as well. So you have volcanoes going off, you have caterpillars that defoliate, you also have, on a longer scale, you have glaciation events because, of course, it's montane. So the question is, how on earth has this tree species actually survived through all of this to become one of the most common species in this part of the world? Is it local adaptation? And if it is, what is actually driving it? This study is based on the GenTree project, which you'll probably hear more about in this conference. Essentially, the methodology is very, very similar. And GenTree looks at 7 species in Europe, but we're focusing on South America. So the first similarity is we use the paired site study design from the Lauterhaus and Whitlock study of 2015. And the idea here is that you're trying to improve your ability to find like SNP outliers by minimizing the neutral genetic differentiation while improving the environmental differentiation. So essentially, you're looking for like 2 sites that they share an evolutionary history but are in different environments. So what this looks like on the ground, here's an example from the southern part of the range near the ice fields. And so these 2 sites are about 1 kilometer apart, but they're over 300 meters in elevational difference. And this, we always try to have at least 200 meters of elevational difference just to get that assured temperature differentiation. If you zoom out, this is kind of what it looks like in the entire range. So we have each one of the black dots is a site pair. We intended to have 9 of them. We ended up with 8 due to some geographical constraints. And then in addition, we had 2 planned xeric sites. So these are the red dots in the center. One is kind of out in the steppe by itself, very, very dry. The other is up in kind of a blind valley in the Andes. It's very high and dry. The third unplanned, unpaired site is this furthest north site. And essentially what happened is we got to the forest and realized, oh crap, there's only 1 elevation we can actually sample at. So we did 1 site there, and then we moved the intended pair to the center of another pair that already existed. So we had a total of 20 sites, 17 of which were paired, 3 of which were unpaired. At each one of those sites, we tried to get 25 adult trees. We had them at least 30 meters apart to try to minimize the familial relatedness, and then we tried to get trees that were at least 50 years old. At one site, we only had 21 adults. So we had 496 adult trees, and from each one of those, I took some DNA samples as well as some dendrocores for phenotyping analysis later on. The first analysis that I did was with the genetic information. So in this species, there has been some genetic analyses, but mostly with microsatellites. So this is the first one of its kind to use SNP data. The starting resources we had were an RNA transcriptome with contig information from our partners in Argentina. We also used the exact same list of candidate genes from GenTree, so it's 1,789 candidate genes that were identified for these 7 species in Europe, because we want to also look for convergent evolution. And then in addition, we added 400 additional genes specific to this species from our partners in INTA, essentially related to things like circadian clock, wood growth, and similar things. And then we did targeted sequencing for those genes. The results we got out of that, we had about 50,000 SNPs after doing some genotype and quality control. After further filtering for things like minor allele frequency, paralogs, we had about 13,000 SNPs. And for you guys in the audience who are GenTree members, we had almost 1,600 candidate genes of overlap. So even though Nothofagus is a Gondwanan species, so it's split from Europe a very long time ago, there is still quite a bit of genetic overlap. The first thing I did with the genetic data is a population structure analysis, so running admixture. I used everything from K1 to K20 because we had 20 sampling sites. It looks like K3 or K4 is probably the most likely based on CV error as well as previous information we have from our colleagues in INTA. And you can see it lines up latitudinally, so probably there's a northern, a central, and a southern ancestral population. The other thing I'll point out is in K4, the northern site here, that one right there, so that always pops out. Everything from K4 to K20, it's always by itself. And in further analyses, things like FST, PCA analyses, this is always, it's a very strong outlier. And so for the rest of the analysis in this presentation, I actually removed those from analysis just to get some clearer results. For the environment data, we used the CHELSA dataset because there's not that much on-the-ground information in Argentina. CHELSA is essentially, it's kind of like this cornucopia of environmental variables that you can choose from, which is nice, but also you have to be kind of careful about which ones you actually do choose. So it has to be something that's representative of the environmental conditions we want to look at, meaning they probably are likely to affect the candidate genes that we selected. So for example, when we look at phenology, looking at like flowering time, leaf senescence, things like this, you might look at things like day length, growing degree days, or frost days, but you have to pick one of those because they also can't be strongly correlated. So we're still kind of in the process of picking the correct variables. And for the rest of the presentation, I give the baseline, so like the bare minimum of environmental variables that I'm going to use for the study, and then I'll probably add some more later on. So the first thing I did, combining the genetics and the environment data, I did environmental association analyses. The first one was a latent factor mixed models, which is a univariate approach. Essentially, this looks at one SNP and one environmental variable at the same time. And for the kind of basic analysis, I ran 3 bioclims that were fairly uncorrelated with each other, so isothermality, max temperature of warmest month, and precipitation of driest month, looking at temperature, precipitation, and then average day length in January, which is related probably to circadian clock and phrenology. So this incorporates population structure data, the latent factors, so I used K3 from the admixture results. I used a fairly lenient false discovery rate of 0.05, and again, I removed population 1. In this analysis, we had about 55 outliers, 39 of which are unique, but again, this may change as we add other variables. I also ran a redundancy analysis, and this is a multivariate approach. So this looks at multiple SNPs and multiple environmental variables at the same time. And the good thing about redundancy analysis is it might pick up SNPs that are under weak selection, which is generally true for things like polygenic traits or things that are just not picked up by other analysis methods. I used a slightly more stringent false discovery rate for this one, 0.002, based on some recommendations I found in literature. However, the population structure is currently unaccounted for in this analysis, so I might have to fiddle with that a little bit. But in this example, I used the same 4 environmental variables as I did in LFMM. The graph here, every colored point is a SNP outlier, and then the color represents which environmental factor it correlates most strongly with. So it's not the only one, but it is the most strong. And the general, so like the standard operating procedure for doing an outlier overlap analysis to look at multiple analyses, and then the more places that SNP shows up, the more evidence you have that it's probably a true positive. So in addition to the environmental association analyses that I mentioned already, the LFMM and the RDA, I also did some that are just looking at the genetics, so looking for outliers just in relation to the underlying neutral genetic structure. I use the same false discovery rate of 0.05 for this one. And I specifically use outflank and PC adapt, which are two methods used in R. So the first thing I'll point out is the RDA actually had the most number of outliers, which indicates we might have a lot of SNPs that are weakly selected for or a lot of polygenic traits. The overlap in all 4 of the analysis was 10, so this is kind of the strongest evidence we have that it's probably a true positive. And then overlap in at least 3 methods was 47. So moving forward with this, I'll take this information, look at those SNPs, cross-reference that with our annotated transcriptome to see which genes these actually came from, and then see which environmental factors those correlate with, and see what is actually driving local evolution or local adaptation in this species. So the next analyses that I'll do, I have dendrophenotypes and candidate gene association. So I'll look at basically phenotype-genotype association, looking at those wood genes again that were supplied to us from our partners at INTA. Also looking at demographic history. So going further with the admixture, kind of looking at the past glaciation events, where were the refugia maybe, how did those populations grow back together after the glaciers retreated? And also looking at gene flow using SSR data. So there's not much information currently in this species related to gene flow. So eventually, we'll take all of this information, so the things I presented today, these 3 things here, and kind of put them all together to paint a better picture of local adaptation in this species. I'd like to extend a huge thank you to everybody who's helped with this project. It's taken a village to even get this far, field work and everything. So my supervisor's here today, my supervisor's watching from Argentina, and also many of you in the audience have helped with this in some way, either by teaching me methods or moral support or answering questions. So thank you very much, and I hope to continue that collaboration. And questions?